Good afternoon, good morning, or good evening, depending on where you are in this world. I would like to welcome you to the 19th Annual Wiley Prize in Biomedical Sciences. And I am Deborah Wiley, Chair of the Wiley Foundation. This program is kindly hosted at Rockefeller University as part of their Friday afternoon series. Over the course of the last 214-year history, Wiley has enjoyed a productive and successful partnership with the scientific community for the advancement of research. We conceived the Wiley Prize in Biomedical Sciences as a way to recognize and to give back to that community. Wiley chose to focus on biomedicine because it is an area continuing to experience significant progress and change. Our goal is to recognize specific contributions or a series of contributions that have opened new fields of research or advanced novel concepts or their applications in a particular discipline and demonstrate the nominees' significant leadership in that field. This year's winners, Brangwynne, Tony Hyman, and Mike Rosen, have indeed made such a contribution. Their work has focused on revealing a new principle for compartmentalizing subcellular organelles based on the formation of phase-separated biomolecular condensates, a process that has implications for both physiological and pathological events. The Wiley Prize jury consists of Tizia DeLong as chair of the jury, Dr. Kai Salicati and Wayne Anderson, both of Columbia University, Joan Stites of Yale, Robert Horvitz of MIT, and Rick Lifton of Rockefeller. This team collaborated to make the selection of this year's award recipients. Tizia DeLong will serve as the moderator for today's lectures, and a Q&A will follow the presentations. Thank you. Tizia?
Thank you, Debbie. And thank you, Debbie, for all the efforts you've put into this award and for the generosity of the Wiley Foundation in establishing and supporting the award. Let me also add my congratulations to the three awardees for this highly deserved recognition of their remarkable work. As Debbie indicated, today's award honors a breakthrough in cell biology, the discovery of how membraneless cellular compartments are formed. The existence of membraneless organelles, often called bodies or puncta, had been known for a very long time. Examples include the Nucleolus and Cajal bodies, but what exactly they represented and how they are formed was not known. This problem was solved by a physicist, Cliff Brangwynne, a cell biologist, Tony Hyman, and a chemist, Mike Rosen, who each synergistically made path-breaking contributions to the discovery that membraneless organelles are liquid-liquid phase-separated entities, which I will refer to as molecular condensates. The large and blossoming field of biomolecular condensate was initiated by two completely independent discoveries. The first was the finding by Tony Hyman and his then postdoctoral fellow, Cliff Brangwynne, that P granules, which are membraneless organelles in C. elegans embryos at the one cell stage that determine where the germline will be formed, showed behaviors such as fusion, dripping, and wetting that suggested that there are liquid-liquid phase-separated droplets. While Hyman and Brangwynne were poking their baby worms, Mike Rosen discovered the formation of liquid-liquid phase-separated droplets when he mixed together two specific proteins in vitro. This unusual droplet formation was manifest when he mixed a protein with multiple SH3 domains with a protein with multiple proline-rich motifs, which is what SH3 domains, which function in cell signaling, bind to. He went on to show that these two proteins together form puncta, these dots, in vitro, and that such behavior was relevant to the behavior was relevant to the signaling that controls actin filaments. So Mike concluded that low affinity interactions, such as those between the SH3 domain and the proline-rich motif, between multivalent proteins or RNA could induce liquid-liquid phase separation, both in vitro and in vivo, just like what was observed by Hyman and Brangwynne. The principle that multivalent low affinity interactions between select sets of macromolecules, some containing intrinsically disordered region, form biomolecular condensate with unique dynamic properties is now well established. And in recent years, hundreds, if not thousands of papers, perhaps not all of them equally rigorous, have implicated this concept in almost every intracellular process, including RAS signaling, immune synapses, DNA repair, transcriptional activation, and the function of nuclear pores, the nucleolus, and centrosomes. Since their initial discoveries, all three awardees have continued to push the field forward, and they now will each give a short talk on their work. I will briefly introduce each speaker and then moderate the Q&A after the third talk. Please use the chat option in Zoom to type in your question and indicate which speaker you would like to hear. Once we start the Q&A, I will call on you by name so you can unmute and reveal yourself and ask your question. So Cliff Brangwynne is our first speaker, and I believe the first Irish recipient of the Wiley Prize. He received a PhD in applied physics from Harvard, working on microtubules. When he was a postdoc with Tony Hyman, he used the principles, concepts, and language of physics to describe the liquid-liquid phase separation of the C. elegans P. granules, and soon after, with Tony, also the Nucleolus. After his postdoc, Cliff joined the faculty of Princeton, where he is now an HHMI investigator and a recipient of the MacArthur Award. Combining the concepts and tools of soft matter physics with molecular and cell biology, he continues to have a major impact in the field. Recently, he showed how the nucleus actually contains two layers with different miscibility, forming a droplet within a droplet. He also managed to engineer a nifty light-inducible tool for condensate formation, and then combined these opto droplets with Cas9 targeting to create condensates at specific genomic loci. You will hear about these and other marvels from Cliff's lab in a talk entitled Intracellular Phase Separation, Physics and Cell Physiology in Disease.
Thank you, Titia, for the wonderful introduction, and Debbie for her hard work with the Wiley Foundation. I think this is an incredible honor for me, and I'm tremendously honored to be recognized along with Tony and Mike for the work that we've done. I think is obviously an interesting time in the world right now, and I think, given everything that we've all been through over the last year, the importance of science to society has never been more crystal clear, and recognition for work going on in science from the Wiley Foundation is I think, really essential and important. And so I'm greatly honored to be here and tell you about the part that I've played in this particular scientific adventure. So I thought I would give you a bit of a historical walkthrough and how we got to work on this problem. I was an undergraduate at Carnegie Mellon University in Pittsburgh, and I decided to study material science and engineering after taking the introductory MSc course, where we poured molten iron alloys into a crucible, and we watched how it crystallized and formed this beautiful dendritic growth structure. And I was really amazed by how the atoms in this molten metal somehow knew where to go. They knew how to snap together just right into this crystalline organization, and that was something that was really remarkable. And I spent my undergraduate time studying the physics of materials. But in this same time period, I took a year off in the middle of my undergraduate and worked at Harvard Medical School in the lab of Don Ingber, and really fell in love with biology at the same time, and started to become interested in related sets of questions, in this case, how biomolecules know where to go and what to do in the structural organization that makes life work. So this is a problem that has been of great interest to me for some time, and thinking about the cell as a kind of living material. For my PhD, as Tizia mentioned, I studied at Harvard and worked in the laboratory of Dave Waits. Dave was a pioneer of soft condensed matter physics, which is essentially the physics of squishy materials. And so some of the work that had been going on in the Waits lab for many years involved colloidal assemblies, things like colloidal aggregates, colloidal phase transitions into these crystalline solids, and gelation, the gelation behavior that happens as they start to get together and interact. There's also a lot of work going on in oil-water emulsions, developing engineered emulsions using microfluidic devices that have lots of applications, and thinking about the rheology of complex fluids, things that many of us know, for example, in silly putty, that has this interesting flow behavior. It acts like a solid under some conditions and a liquid under other conditions. But in the Waits lab, I was thinking about these links to cell biology and was actually working in this environment, but on living cells. And in particular, I was studying microtubules and trying to use microtubules as probes of the mechanical and rheological behavior within living cells, essentially little calibrated rods that we could use to understand the fluctuation dynamics within living cells. And so, so trying to put these ideas together, some of the concepts of soft matter physics, is something that I've been quite interested in. Around this time period during my PhD, I had the great fortune of taking the famous physiology course at the Marine Biological Laboratory. So this was in 2006, towards the end of my PhD, and I didn't circle it yet. I can let you, scan the image and see if you recognize anybody. That's me in the back. This was an important moment for me because the physiology course gave me an opportunity to see biology in all of its glory and kind of the mind-blowing beauty and complexity of biological systems. It was also really important for me as a moment because it was where I met Tony, who I later did my postdoc with. And so that was an important connection. The physiology course had been started by Tim Mitchelson and had been adapted or evolved under the leadership of Tim Mitchelson and Ron Vale. Tim is shown here. Tim is actually Tony's postdoc advisor, so there's an interesting connection there. And Ron, you see. So Ron and Tim had taken over the physiology course and brought together physicists and biologists in this quantitative biology program. So it's really an exciting place for me to be at that time and see the connections with biology. So I decided to do a postdoc with Tony in Dresden and became really interested in these structures that Tizia already mentioned. These are called P granules. Related structures are found in essentially all animals, or go by different names like nuage or germplasm. They have a role in specifying the germ line in development, and in this case of the C. elegans embryo. They're RNA and protein rich, so lots of RNA and lots of protein, and they have no enclosing membrane. And that was one of the things that I think was, to me, just very fascinating. Somehow these are coherent bodies with many hundreds or thousands of molecules, and yet there's no enclosing membrane. They're part of a broader class. There are dozens and dozens of different types of membraneless organelles, what we now call condensates, intracellular biomolecular condensates. This includes a lot of things in the cytoplasm, like stress granules, processing bodies, and signaling assemblies, and many structures in the nucleus, like nuclear speckles and nucleoli, which I'll tell you a bit more about, and Cajal bodies. So again, as Tizia mentioned, these are structures that were ubiquitous and in many cases known for a long time. The Nucleolus was identified in in cells, 2 centuries ago. And yet it was not clear what held these structures together, what were the physical principles that held them together. So it's a really interesting problem that I started thinking about at this time. And so I worked at the Max Planck Institute for Cell Biology and Genetics, and also the Max Planck for Physics of Complex Systems, working closely with Tony, Christian Ekman, who was another group leader in the MPI CBG, and Frank Ulischer, who was the director of the MPI PKS. And the specific problem we started tackling in the beginning was trying to understand how these P granules, shown here in green, how they localize to the posterior end of the embryo, shown on the right. This was a really interesting problem, and we were initially just thinking about it as a transport problem. How do these structures get to one side of the embryo? And through some quantitative imaging, three-dimensional particle tracking, confocal imaging, we were able to show that P granules on the anterior side, on the left side of the embryo, just steadily dissolve in time. They just kind of shrink and shrink and shrink until they're gone. And P granules on the right, on the posterior end of the embryo, initially are dissolving and shrinking, but eventually they're stabilized and actually grow. So there's a spatial gradient in the stability of these structures over the course of the development of the embryo. And so we were wondering, what causes this, and how to think, what's the right physical picture to think about this spatially regulated stability. Now, right around that time, and this is now the summer of 2008, I went back to the MBL with Tony as a teaching assistant to teach in the physiology course. And this was also an important time. It was exciting time. We brought the P granule project and had students looking at this process. And this was in the course of this 2 weeks that we're at the physiology course, made some observations that got us thinking about these structures maybe behaving a bit like liquids. We were making movies and we saw them fusing, and started wondering if they were perhaps behaving like liquids. That turned out to be the case. So this is a movie that I took soon after, got back to Dresden. This is a very low budget rheology experiment where we essentially squash the worm, do a dissection of the gonad of the worm and squash it, and do shear stresses. And what you see is that this P granule material flows and drips and wets, coalesces, much like conventional liquids. So this was an exciting time. We at that stage, we didn't quite know what it all meant, but we thought this is really interesting that there are these structures that are behaving like liquids in living cells. And what was particularly compelling about it was that it fit together very nicely with the observations of the spatially regulated dynamics. And so we put that together and put forth this model wherein the P granule segregation process is a spatially regulated phase transition, what we call liquid-liquid phase separation, wherein the condensed form, the biomolecular condensate that forms in the posterior end of the embryo is stabilized, whereas in the anterior end of the embryo it's destabilized, and essentially it's undersaturated in the anterior and it's supersaturated in the posterior. So this was really, again, an exciting time. It was exciting to think about these liquid states of matter in living cells. We're all familiar with liquids from everyday experience. I think everybody has some intuition for what a liquid is, from salad dressing, which is a kind of a demixed liquid-liquid phase separated system, oil and vinegar. But we can think about liquids and dewdrops on grassy morning, or teardrops on a wine glass, and so forth. Liquids are also interesting from the perspective of the physics at play in their properties. And it turns out there's still a lot of ongoing research in the fundamental physics underlying liquid behavior, things like this Rayleigh plateau instability or the surface tension coalescence behavior of liquids. There's ongoing research programs even in non-living matter. Liquids are also important and interesting from an engineering perspective because they're useful in many applications. These are some examples of microfluidic emulsions that are made and have applications in RNA single cell sequencing and cosmetics and many other applications. So it was exciting to think this was relevant, this kind of physics was relevant in living cells. And we at this stage wanted to know whether it was general, if it was something specific to P granules or more general. And so I went back to the MBL again, this time to work in the summer laboratory of Tim Mitchelson. And with the help of Tim and together with Joe Gall, I took a trip down to the Carnegie Institute to learn how to use the frog oocyte system from Joe Gall. We were able to study these nuclear bodies in the nucleus of this of what's sometimes referred to as a germinal vesicle of the frog oocyte. And within this nucleus, there are hundreds of nucleoli, which I'll tell you a bit more about, but are really interesting nuclear bodies. And we were able to do some really simple experiments to show that they behave like liquids. They coalesce with one another. We can pull them apart, and there's liquid bridge rupture that occurs. And so this system represents an emulsion of biomolecules that have condensed into these droplets and phase separated out. What was interesting about this system is that it's a kind of active emulsion. We were able to show there are ATP-dependent processes that change the properties, the viscosity and surface tension in the frog oocyte. So my lab at Princeton has been really interested in a number of different condensates throughout the cell. We now understand that there are dozens and dozens of different types of phase-separated condensates throughout the cell. We've been really excited about what's happening in the nucleus, phase separation in, on, and around the genome. Essentially, these structures as little dewdrops on the genomic landscape. And we've continued quite a bit with the Nucleolus, which is the largest of these structures in the cell. And to me, at least, the most interesting. It assembles around sites of nascent ribosomal RNA transcription, and so it facilitates the transcription and processing of those ribosomal RNAs to make functional ribosome, ultimately forming these pre-ribosomal particles. So we've been able to show, my graduate student Marina Ferrick did these beautiful experiments and showed that the Nucleolus represents a multi-phase liquid, essentially a liquid within a liquid that we think is important for the sequential processing of these nascent ribosomal RNA transcripts. We can actually recapitulate that liquid within a liquid multi-phase behavior in vitro using purified components. This is interesting for me as well because it's very reminiscent of the kind of multiple emulsions that the Waits Lab had been producing around the time of my PhD. We've been really interested in how these kinds of this organization facilitates and is connected to the transcription and processing of these nascent ribosomal transcripts. Some of that work we had been doing in the early C. elegans embryo, which continues to be a very powerful system, and watching the assembly and coarsening dynamics of nucleoli as the nuclear envelope reforms. And we've been able to connect those dynamics to some of the fundamental physics that one would predict from a phase separation picture, in particular building from Cahn-Hilliard frameworks and other models from materials physics. More recently, we've been able to show that the thermodynamics of the Nucleolus facilitates the vectoral flow out of the Nucleolus of these pre-ribosomal particles. As the transcript first is transcribed, it's binding with all of the other proteins that are in this liquid matrix, if you like. And then as the ribosomal RNA folds and binds together with ribosomal proteins, it becomes strongly thermodynamically unfavorable for it to remain in the Nucleolus, and so it's essentially ejected. Once the structure is made, it's all done and leaves. So those are some of the things that we've been trying to do to connect the fundamental physics with the functional outputs of these kinds of structures. Another thing that we've been quite interested in for some time is this connection between the soluble, or what you might call a gas-like state, the soluble molecules, where and when exactly they transition and condense into the liquid states. And the hypothesis has been for quite some time, we put forward this idea in a mini review in 2012, that these liquid states can transition into solid states, which might be pathological. So just like water, we can put water in the freezer and it undergoes a phase transition into a solid state. We think that these liquids can transition into solid states that are by and large pathological. MBL shows up again in this narrative. We went back to the MBL In 2014, I brought a couple of students from Princeton, and this was part of the Summer Institute that Mike and Ron Vale and Jim Wilhelm had set up to study phase separation, intracellular phase separation. So this is an exciting collaborative environment. You can see me again, the tall guy in the back, and Mike. Probably recognize other folks like Amy Gladfelter and Ron Vale. So this was a place where we started to test some of these hypotheses. In particular, a couple of papers that I think are influential came out of that Summer Institute in 2014. Mike, together with Roy Parker, had a nice paper on showing HNR and PA1 forms these liquid droplets in vitro, but those liquid droplets age in time and solidify. And work that we did collaboratively with Amy Gladfelter that grew out of that summer, worked on this protein WE3, which is a poly-Q protein, showing that it also ages and undergoes a subsequent solidification over time. Around this time, there was a beautiful paper that came out of Tony's lab, Tony and Simon Alberti in Dresden, showing that FUS, which is an ALS-related protein, forms beautiful droplets in vitro, but those droplets, again, age in time, and that aging is linked to mutations in FUS that are genetic mutations found in familial ALS. And so really interesting connection between liquid-liquid phase separation and subsequent solidification that may be associated with disease. So I think Titia also set us up nicely here. There's many important condensate proteins contain both IDRs and oligomerization domains. The IDRs are intrinsically disordered regions. And so prior to this whole development of this field, most people would have viewed disordered proteins as associated with pathology only in aggregation. What we now know is that these disordered regions are key for the assembly of these physiological condensates, things like the nucleolus and stress granules and many other structures in cells. These are some of the examples from my lab where we've purified proteins that have significant disordered regions. In many cases, the disordered region is necessary and sufficient for driving phase separation in vitro, and we think it's driving, in many cases, the corresponding structure in cells. What we've realized even more recently is that one of the key domain architectures that's commonly seen in key structural proteins for condensates includes oligomerization domains. And oligomerization is really an essential aspect of the driving force for phase separation. So polymer physics, going back to Paul Flory 70 years or so, we have this framework where we can understand how the degree of oligomerization or the degree of polymerization can drive phase separation. Essentially what it's doing is it's acting as a kind of entropic sink, so decreasing the entropic driving force, which generally wants things fully mixed, not phase separated, such that those interactions between the biomolecules can put the system over the phase boundary. And I think you're going to hear more about that from Mike in the context of these multivalent signaling proteins that he's been studying and has done really pioneering work in. So one of the things that we've been really excited about is using building from these native oligomerization domains, and in some cases we can just swap out the native oligomerization domain with an engineered oligomerization domain that is light activatable. So when we turn the light on, we turn on oligomerization, and when we turn off the light, we turn off oligomerization. And this has been really, really interesting because when we bring intrinsically disordered protein regions together in these oligomerized states, mimicking what's happening endogenously, they phase separate. And so here are cells, these are U2S cells. We're expressing the disordered region of this FUS protein. And then when we shine light on the cell, we get droplets that form, in this case, throughout the nucleus. We can also do it in the cytoplasm and other places. And these are very clearly phase separated liquids mediated by the disordered regions of these proteins. We have all kinds of fun with this. We can cycle it in time and ask about reversibility and interrogate these connections to irreversible subsequent solidification transitions. And because it's light patterned, we can draw pictures in cells and put smiley faces, things like that, wherever we like. That's actually been important for asking about the functional consequences for these condensates in different places in the cell. What's also been really something that I'm very passionate about is being able to quantitatively map phase diagrams within living cells. And these optogenetic approaches that we've been pioneering, we've been developing, allow you to do that. And essentially, this axis is the oligomerization state. Because you're essentially turning entropy up and down, you can think about this as a temperature axis. So higher the higher up the axis you go, the lower the oligomerization state, the more entropy matters, just like if it was a temperature axis. This is a core concentration, so the concentration of the molecules. And what you see is within this two-phase region, these are concentration values where the cell phase separates, and these are concentration and oligomerization values where they don't. So we can quantitatively map the threshold, the phase separation threshold. And this is also interesting because we can see behaviors like nucleation and growth, spindle decomposition, gelation behavior deep into the two-phase boundary. This, for me, has been interesting because it actually takes me back to how I got interested in these kinds of things way back in the beginning of my undergraduate days, where we were studying the phase behavior of materials like steel. This is actually from some notes from an undergraduate course I took, where we were looking at the phase boundaries of steel. And so we're essentially doing the same type of thing now, except we're using these approaches to interrogate, to forward and reverse engineer liquid states of matter in living cells. We're essentially pouring biomolecular condensates into cells and asking what the consequences are, and asking if we can do useful things with those technologies. So I want to thank you for your attention. I tried to mention folks that have been part of this adventure. Of course, I didn't have a chance to mention all the amazing students and postdocs in my laboratory, and current and former have gone on to bigger and better things, and a lot of the collaborators we've had. I'm not going to go through the full list, but obviously science is a team sport, and being recognized with this award is incredible, and it's an incredible honor. But I would also emphasize that science really is a team sport, and we've stood on the shoulders of many giants and worked together as a team to get where we are, and I'm grateful for the opportunity to work with many brilliant people in that effort. So thank you so much.
That was a terrific overview. It's very cool experiments. Just to remind you, the Q&A will be after the third talk, and I want to apologize for my throat. It's spring, and pollen are in the air. So our next speaker is Tony Hyman, who has been a Max Planck director in the beautiful Dresden since 1997. When he and Cliff first observed the liquid-like behavior of pea granules, Tony was already widely acknowledged as a highly creative leader in the field of cell biology. After he did a postdoctoral research period with Tim Mitchelson at UCSF, working on the way yeast kinetochores interact with microtubules in vitro work I watched from close by, he went to Germany, first to the EMBL in Heidelberg, and then to the Max Planck in Dresden, bringing his interest in cell division to the C. elegans system that he had worked on as a graduate student with John White in Cambridge. Tony made major advances in our understanding of how centrosomes and microtubules work together to form the spindle that orchestrates the segregation of chromosomes. Having a deep and broad appreciation for cell biology, it's no surprise that Tony immediately saw the importance of what he and Cliff observed. And in the past 10 years, he has not only continued to make major discoveries in phase separation, including the interesting finding that his beloved centrosome is a condensate that promotes microtubule formation. But he also became an eloquent and inspiring spokesperson for the field. And he will now tell us about what he calls a journey in C. elegans cell biology.
Thank you so much, Thetia, for that lovely introduction, and also Deborah and the whole Wiley team for making this work. As Cliff said, it is an incredible honor to be awarded it with Cliff and Mike, who spent the last 10 years, all of us together, really working on the consequences of these discoveries. So I'm sorry we're not all together, but it's a great pleasure to be giving this talk today. And my journey actually began when I was an undergraduate. I had an incredible cell biology course at University College London, where I was introduced to all of these membreless compartments in cells. But although it was clear that the cell was full of these bodies, no one knew very much about how they formed, or a lot of the functional roles of these bodies. And yet we now estimate that 50% of proteins in the nucleus are probably in such compartments. I mean, it's a really complex problem, because if you think of like a nucleolus that Cliff mentioned, the site of ribosome production has no membrane surrounding it, hundreds of proteins required to make the ribosome. And yet, without a membrane, how does that biochemistry work in one place? So if we sort of look at that problem with a sort of modern understanding of protein dynamics, basically molecules will be diffusing around in the cytoplasm at, say, 5 square microns per second. And then these bodies tend to be very transient. So a signal might come in of biochemical need. Then the bodies come together to form these membreless compartments. And then when they're no longer needed, will dissolve and the proteins will start diffusing around again in the cytoplasm or the nucleoplasm. So let's think about that problem then quantitatively, because let's take a typical membreless compartment such as a stress granule. If you roughly what we know now of the concentration of proteins in the cytoplasm, you might end up with 1 million molecules inside a stress granule. Inside a nucleolus, it might be 25 million. But 1 million is roughly the size of a small city, such as Dresden. Now, a cell has about 5 billion proteins, which is roughly the population of the world. And so in principle, you could imagine a dilute solution of people, and they all come together to form a city when they're needed, and then they disappear. But actually, the cell is a more bizarre place than that, because if you take proteins and scale them up to be people, and kept the same density as in the cell, we'd all fit into the Great Salt Lake. Arm separate about arm to arm, diffusing around about 30 square kilometers a second, not making inappropriate interactions, but when necessary, forming these compartments to do biochemistry and then dissolving. So this is an incredibly fascinating problem, and it's really occupied most of my life in trying to understand how it works. Now, my journey scientifically began when I started a PhD at the LMB in Cambridge, and I ended up in John White's office, which was lucky because everyone else was working on DNA, and I turned out not very interested in DNA. But John pointed out to me that Victor Nigon in the 60s had introduced C. elegans embryos as an incredible system to study cell division. And he suggested to me that I could look into, in the classic British supervision style, how compartments and spindles could form in these developing embryos, and also encouraged me to work on my schoolboy French to read the classic paper. At the same time, he sent me to the library, and I found this book, E.B. Wilson, which had summarized in 1920 this cornucopia of incredible cell biology, often described membreless compartments that no one had seemed to work on very much since then. And I just became very fascinated with trying to understand that problem in C. elegans embryos. For instance, one of the things I noticed when I was a PhD student, this incredible array of microtubules that form in C. elegans prior to mitosis. And I became fascinated in understanding how they might form. So next I went to do a postdoc with Tim Mitchelson. As Cliff mentioned, I think Tim has been a real thread through much of our work and a great mentor. And I learned quantitative biochemistry with Tim, which has been so important for later work we've done on reconstituting compartments in vitro. And that was a wonderful time. I met Tizia. And then the first group was at EMBL. And I did many things there, but probably the most important thing was I met Eric Carcenti. He has an incredible beard, as you can see, who's gone on to run this Tara Oceans project. But Eric, at that time, was getting interested in the role of physics in studying biological problems, because he's working with Stan Leibeler, as you know, went off to Rockefeller. And he started to explain to me that we could really think about using physics. I mean, I was a cell biologist, actually a zoologist, really. So it was all pretty, but I come to understand how useful it could be. And that was stimulated by the chance I had in 1997 to go with a founding team to Dresden after the wall came down. It seemed like a sort of crazy idea in retrospect, but it's very hard to resist the charm of Kai Siemens for anyone who knows him well, and to build a new institute. And of course, as a Max Planck, you can choose what you'd like to do. And there we decided one of the things that Max Planck had done was to build another institute, the Physics of Complex Systems, and hired Frank Jülicher. And together, Frank and Joe created this sort of physics multidisciplinary environment in Dresden. And we settled down to think about how we could use physics to think about the formation of the C. elegans embryo. And, you know, over the years, we did some interesting experiments. We worked on the collective properties of motors. We worked on reaction diffusion systems in the cortical assembly. But in a way, all of these were taking physics that had already been used in cell biological systems to understand some of these problems. And the question of how cells form membranous compartments has sort of gone rather dormant until my paths crossed with Cliff Brangwynne in physiology course. And my sort of telling of that origin story is that Cliff was taking the course when I was one of the instructors the year before. And I had a rather studious bunch of students. But next door was the fun side. There was always laughter happening in the other group. And every time I went across, there'd be Cliff in the middle of these students and postdocs, you know, lots of laughter happening. So, you know, I got to know Cliff that way. And when he came to Dresden, of course, it was you know, very happy that, and he wanted to study this problem of C. elegans P granule formation. And that was very interesting in a sense, because actually Susan Strohm and Bill Wood had discovered these P granules when I was a PhD student. It was incredible excitement, but actually in the intermediate time, really come to understand how they segregated and what the actual cell biological mechanism might be. And it was during those Woods Hole years that Cliff, as he mentioned, made these incredible observations about the fact that they're liquid-like. Here are two movies showing P granules fusing. At the time, we proposed that they are liquid-like and they form by phase separation, a little bit like oil and vinegar separate in a vinaigrette. We went on, as did others, to show that many compartments could be described as liquid-like. This is one in the stress granule, work we've done with Simon Alberti, showing that the stress granule is very liquid-like. You can see it kissing and fusing and relaxing into a drop. And so, as Cliff mentioned, that's become more and more, you know, it's clear that more and more compartments can be described as these phase-separated liquids. So you can ask the question, why were we so excited at the time about this? You know, what got us excited to begin to think about it more? The reason was that thinking about these compartments as phase-separated liquids allowed us to understand problems that have been recalcitrant to our studies previously. And you know, Cliff mentioned one thing, which was the segregation of P granules as a gradient of condensation. But let me show you another example, which is very dear to my heart, because I mentioned how as a PhD student, I come across these microtubule dynamics and how these incredible nucleation of microtubule structures. And I kept working on that problem without much success, actually, since then. For instance, gamma tubulin, which was a known microtubule nucleator, we did a lot of effort with Karen Ugemer to remove it genetically, but still we formed these microtubule asters. We even did these genome-wide screens at the turn of the century with Pierre Guncy, where we actually RNA'd every gene in C. elegans and did a videotape of the movie. And still that didn't really show us how microtubules nucleated. Now, there could be a number of reasons for that. But once we thought about phase separation, we began to think about this problem in a different way. And that's for the following reason. One, the crucial aspect of phase separation is you can think of the density transition, right? In vitro, you can get up to 1,000-fold concentration by phase separation, but it's still a liquid. The molecules are still moving around fast inside this liquid structure. And of course, once you think about concentration, you can imagine the concentration of biochemistry could accelerate reactions. Now, why is that? Well, many people have done this experiment, even probably in high school, which is a Michaelis-Menten enzyme kinetic experiment. You take an enzyme and you increase the substrate concentration and the reaction velocity goes up. Now, we hadn't really thought about that in terms of cells, because how would you increase the concentration? Rather, we'd focus mostly on post-translational modification, which is, of course, very important for regulating cellular behavior. But this suggested we could think about concentration as a way of regulating activity reactions. And to show you that, let me just tell you one thing about microtubules that's important. This is a graph of tubulin concentration against absorbance. And you can see you have a critical concentration. So at 5 micromolar, there's no nucleation. And we go over to, say, 30, and now you get nucleation. So that suggests if you raise the tubulin concentration, you could actually trigger nucleation of microtubules. Now, just the experiment I'm going to show you requires an understanding of something that Mike introduced, this concept of scaffolds and clients as an operational definition that's been very helpful in the field. That are a few scaffolds that form by phase separation and many clients that then partition into their scaffolds to drive the enzymology. So here's the experiment. We took tau, which is a microtubule binding protein, added a little bit of crowding agent, and we got this drop of tau, beautiful liquid. Now, that's the scaffold. Now we add the client, which is tubulin, and it's partitioning into the drop. So here's the experiment. You've got, in the light blue, you've got 2, 4, 6, 8 micromolar tubulin, but the dark blue shows it being incredibly concentrated inside the drop, much more than the input tubulin. So now we've raised the concentration and we add the nucleotide, and now we see the formation of local microbundles, driven, we believe, by the local increase in microtubule concentration. Now, that was, when we saw it, pretty spectacular. Of course, it's a toy experiment, but it encouraged us to go back again to something we never would have done before, which is actually measure the soluble tubulin concentration in a C. elegans embryo. And that's something, in a way, had been known that there's a lot of soluble tubulin in a centrosome, but we actually measured it by electron tomography and other methods, and we came out with an incredible concentration of 400 micromolar, right? Plenty tubulin to drive nucleation, suggesting that concentration of tubulin by partitioning into a phase-separated condensate of a centrosome could be one of the crucial ways of driving nucleation. So just give you that as an example of how the discovery of these phase separators changed the way we thought about so many problems in cell biology, and many, of course, are now being published in the journals as we speak. So I'd just like to finish by giving us a little historical viewpoint of physical chemistry and cell biology, because actually, if you look historically, as we started to do once we made these discoveries, in the 1910, because of the discoveries in physical chemistry, the scientists at the time thought it must be important in understanding cellular organization. Hopkins was this famous lecturer in 1913. He discovered vitamins, that a cell is a particular dynamic state of a polyphasic system. But if you read the subtext of his lecture, it's like, there are too many physical chemists in this room. We don't know any molecules. It's impossible to test these ideas. And so over the years, people started to obviously go down the structural route, and electron microscopy, especially in the 50s, sort of took us away from this liquid-like idea of cells, until there's this amazing quote from Peter Medawar in 1967, saying anyone working on the physical chemistry of the cells is considered slightly mad. So I think that basically what we've done is to bring back physical chemistry in the context of the organic chemistry that sorted out the molecules, and allowed us to understand so much of the questions of regulation that we couldn't understand before. And I think moving forward, the crucial thing is now to describe the physical chemistry of each of these compartments and ask how it relates to the function. We've shown some definitely can be formed as liquid phase separation, but it's important to go through each one now and ask how we can relate the physics to the function. So I just want to finish then by thanking everyone I've worked with. Just as Cliff said, it's a team effort. This is, I think, all the people I've worked with since I started my lab 30 years ago. It's been a great voyage of discovery, and I wouldn't miss the day of it. We've also had a lot of fun. World hasn't had a lot of fun in the last year, but I'm hoping that pretty soon we'll get back again to having a lot of fun as scientists. And I'd actually just like to finish on a different note, which is to dedicate this lecture to my wife, Suzanne Eaton, who was lost in a senseless act of violence in 2019. And if you meet somebody when you're young and you grow old together, then basically your success is a sort of shared success. And many of the discoveries I talked about today really wouldn't have happened without the enthusiasm, love, and sheer intellectual brilliance that Suzanne always brought to any problem. You know, it's amazing to be able to come home after you made a discovery and have such a keen mind to help you think about it. So thank you so much, Suzanne. And finally, of course, thank you again to the Wiley Foundation for this great honor.
Thank you, Tony. That was a beautiful talk about an amazing journey. To remind everybody, if you have a question, please type it into the chat and I will get to it after the third talk, which is by Mike Rosen, the third awardee and the originator of the term condensate, if I'm not wrong. Mike trained as a chemical engineer and a chemist at Harvard, and then embarked on what became an illustrious career as a structural biologist, using both NMR and crystallography to understand how the cytoskeleton, especially actin filaments, are regulated. Mike initially was across the street from us at Memorial Sloan Kettering, and sadly, we lost him and he moved to UT Southwestern, where he is an HHMI investigator and chairs the Biophysics department. Mike's work always focused on the cellular signaling pathways that control cell shape, mobility, function. And some of these signaling pathways involve proteins with multiple copies of the SH3 domain, the sarcomology 3 domain, which, as I mentioned earlier, led him to his insight that multivalent low affinity interactions between macromolecules can lead to liquid-liquid phase separation. Mike's chemistry background is deep understanding of protein structure, his knowledge of the literature on polymer phase separation, as well as his quantitative mind, allowed him to combine theoretical calculations, computer simulations, and strategically designed experiments to produce a quantitative, clear, and experimentally based understanding of how condensates form and behave. He quantified how valency drives phase separation and specified the difference between scaffold and Klein molecules in the condensates, as Tony mentioned. He also showed how reducing the dimensionality from 3D to 2 dimensions, as occurs on membranes, increases phase separation by orders of magnitude. So I now invite Mike, as well as his parents, to join us during Mike's talk on a journey in structural biology from signaling molecules to biomolecular condensates.
Well, thank you very much, Titya, for that very kind introduction. I want to begin this afternoon by joining Tony and Cliff in thanking the Wiley Foundation for putting together this symposium this afternoon and for this absolutely incredible honor of receiving the Wiley Prize. I also want to thank the Wiley Prize jury for including me in this recognition. It's particularly wonderful to receive this together with my old friends and colleagues, Cliff and Tony. As they've indicated, we've been working together to understand biological phase separation and its role in forming biomolecular condensates for a number of years now. Now, as Titya told you, much of the research in my lab for many years, some of it done right across the street from Rockefeller at Sloan Kettering, focused on application of structural, biophysical, and biochemical tools to understand actin dynamics in cells. And as I'm sure everybody in the audience understands, spatially defined rearrangements of actin are critical to a huge array of cellular processes, everything from cell movement to cell division to movement of bacterial pathogens to tumor metastasis. All of these are driven by the rearrangements of actin in a very precisely defined way in cells. And so we had always been interested in understanding the mechanisms that cause actin rearrangements, but also how those mechanisms are applied in a spatially defined manner. Now, much of our work focused on a family of signaling pathways by which members of the so-called Wiskott-Aldrich syndrome protein family, the WAS protein family, activate the ARP2/3 complex to produce new actin filaments. Now, this is a pathway that is tightly localized in a huge range of cellular processes. For example, you can see here in the leading edge of this expanding neuronal growth cone, one member of the WAS family is localized right at the tip of that structure. Now, WAS proteins are regulated through autoinhibitory mechanisms. There's an N-terminal so-called GTPase binding domain that binds intramolecularly to a C-terminal so-called VCA domain, and that silences the molecule towards the ARP2/3 complex. That autoinhibition is relieved by binding of Rho family GTPases to this N-terminal element. Now, separating the autoregulatory portions of the molecule is a large proline-rich region. And that proline-rich region contains a number of motifs that, as Titya mentioned, are the binding sites for a variety of SH3 domains. And that interaction becomes very, very important in just a moment. Now, much of the early work in my lab focused on structural biology of these systems. And so two of our early structures are that of autoinhibited WASP, shown here on the left, and an activated CDC42 WASP complex, shown here on the right. And we determined these using NMR spectroscopy, actually back when I was at Sloan Kettering. Now, these structures explained a huge amount about how the autoinhibited protein is prevented from interacting with and activating the ARP2/3 complex, and how binding of the CDC42 GTPase unfurls that autoinhibited structure and turns the system on in signaling pathways. Now, we published a paper in 2008 that provided an additional layer of regulation on the system, because what we found then was that monomeric WASP in its active form binds to the ARP2/3 complex with a relatively modest, affinity, approximately 2 micromolar. However, if WASP proteins are dimerized, and often that's mediated by interactions of the SH3 domain, of the proline-rich region with SH3 proteins, the affinity jumps up almost 100-fold, so about 20 nanomolar. And that immediately set us thinking then about oligomerization of this system, because if you think it through, oligomerization in general then should potentiate allosteric activation of the molecule. And we started then studying systems that would oligomerize WASP proteins. And one of the ones that we focused on was a signaling pathway that was initially discovered by my postdoctoral advisor, Tony Pawson, that begins with a cell adhesion molecule called nephrin that in its cytoplasmic tail has 3 phosphorylated tyrosines. Those phosphotyrosines then bind to the SH2 domain of an adapter protein called NCK, which then carries with it 3 SH3 domains. Now, those SH3 domains bind to that large proline-rich region of a molecule called NWASP, which has roughly 9 SH3 binding sites in that central element. And what we reasoned is that the multivalency in this system should drive oligomerization and activate that sort of dimerization aspect of the mechanism. And in fact, this was a system that Tony Pawson had shown, forms puncta at the plasma membrane, and those puncta cause the assembly of actin filaments to emerge from them. So we thought that this would be an interesting system to look at this dimerization or oligomerization effect. And what we found when we first mixed NCK and NWASP together was very surprising. We did form oligomers, but beyond that, we got this sort of weird opalescent kind of shimmery solution that now we immediately recognize as indicating liquid-liquid phase separation. But at the time, it really was not clear at all what we were looking at. And it took us quite some time, actually, to stick that solution under a microscope when we understood what was going on, that in fact, when these two molecules had bound to each other, not only had they oligomerized, but they had phase separated. And moreover, the proteins, when we labeled either one with a fluorophore, became very, very highly concentrated in those phase separated compartments. So we came to understand this through the lens of almost a century-old polymer chemistry and polymer physics. Because what polymer scientists starting in the 1930s and 1940s had recognized is that multivalent entities, if they are interact in non-covalent fashion, will form larger and larger oligomers, polymers, if you'll have it, as concentration is increased. So for example, at low concentrations, there might be a mixture of monomers and say dimers. Higher concentrations will give you monomers, dimers, trimers. And at high concentrations, one can get large oligomers, we can call them polymers. Now, what polymer scientists also realized, again, back in the 1940s, is that as polymers get longer, they become less soluble in solution. And at some point, one can get to a length that hits the limit of solubility, and those polymers will crash out and form a separate phase, often a separate liquid phase, because of the flexibility of polymers. And so putting these two properties together, what we reasoned is that multivalent interactions between proteins will drive their polymerization. That inherently will decrease their solubility and ultimately will promote their phase separation. Now, initially, we began to think of this in the context of actin assembly, because as I mentioned, we knew that through cell biological work from other labs, that when actin is assembled by wasp proteins, it very often does so at these micron scale foci that form at the plasma membrane. And so we thought perhaps these phase separated concentrated structures were sort of fundamentally the same as these foci observed in cells. Now, when Cliff and Tony's paper came out, the now absolutely seminal paper showing that P granules in C. elegans embryo are phase-separated liquids, and you've heard the data now that supported that conclusion. When that paper came out, we became very interested in understanding what are the molecular components of those structures and of other granules, bodies, as they had been called at the time. And what we found is that they are all highly enriched in multivalent proteins, and many of them also in RNAs. And so that got us thinking that perhaps this process that we had observed with our actin regulatory proteins was not merely involved in controlling actin, perhaps it was involved in condensate formation really writ large. And occurred very, very generally in cells to underlie formation of these structures. So that led us to our 2012 paper, where we put forward this model where the concomitant assembly and phase separation of multivalent molecules or macromolecules could fundamentally underlie formation of biomolecular condensates in cells. Now, one of the powerful aspects of this mechanism is that it immediately leads through polymer chemistry into modes by which these structures could be regulated, and it also has important implications for function. So one of the ideas, again, coming from polymer chemistry, is that higher valency will promote phase separation. More adhesive elements in a molecule will allow it to phase separate at lower concentrations. And so if you think of phase separation of NCK and NWASP, we have a trivalent 3SH3 domains interacting with approximately 9 proline-rich motifs in WASP. When those NCK molecules, though, become assembled on the 3 phosphotyrosines on phosphorylated nephrin, the valence in this system jumps from 3+9 up to 9+9. And that suggests for a given concentration of those two molecules, they might start off in a single phase regime in this phase diagram. But upon arraying the NCK, the phase boundary can shift, and those same concentrations will now find the system in the two-phase regime, i.e., it will be driven to phase separate. And so that suggests that kinase activity in cells could be something that could control phase separation and generally assembly of signaling molecules. And one can take that a step further and say essentially that any post-translational modification that occurs in multivalent fashion interacting with various kinds of reader proteins that recognize those modifications could be something that modulates phase separation. And these are ideas that have been observed now in a number of different systems. In terms of function, we examined this signaling pathway from nephrin to NCK, NWASP to the ARP2/3 complex to actin in light of or concomitant with phase separation. What we found is if we do a titration that induces phase separation of these signaling molecules, the activity, the polymerization rate of the system jumps up basically right where we see phase transitions occurring. And so what that tells us is that phase separation enhances actin assembly activity, and moreover, that phase separation then provides a source of nonlinearity to this signaling pathway, but also potentially to many others. And again, that's something that has been observed in various other studies. Now, in the interim, there have been many, many different instantiations of weak multivalent interactions driving phase separation of many different kinds of molecules. We and others have shown that a variety of multi-domain proteins can phase separate. I've talked to you about actin assembly molecules. There's some beautiful work from Minji Zhang's lab in Hong Kong showing that the multi-domain proteins involved in the postsynaptic density in neurons also can phase separate, and he thinks that that process may underlie formation of the postsynaptic density. There's been a huge amount of work done on intrinsically disordered regions of proteins, which are very much analogous to these multi-domain proteins in that they are a series of adhesive elements arrayed on a disordered chain. The only difference here is that with intrinsically disordered molecules, the interaction elements are much, much smaller. So cation pi interactions involving aromatic residues, electrostatic interactions, or small elements of cross beta interactions can function to drive phase separation of these systems. Repetitive base pairing interactions in RNA and DNA molecules also have been shown to promote phase separation of these kinds of systems. In one, I think, particularly interesting example, one can take structures, synaptic vesicles, which are obviously much, much, much larger than any of the molecules shown here. And if one coats them with weakly adhesive molecules, they also will phase separate and create a fluid whose basic unit is not a molecule here, but actually a vesicle. And so all of these ideas actually occur in very much a scale-independent fashion. And I'll come to that in just a moment here, that idea of scale independence. So to jump forward a little bit in the last couple of minutes here, what I want to tell you about is an area that we've become recently very, very interested in. And I'm going to set up the area first and then tell you how we got into it through this lens of multivalency-driven phase separation. And that area is the organization of chromatin in the eukaryotic nucleus. So the basic problem here is that every cell in the human body has approximately 2 meters of DNA that has to be compacted into a 10 micron diameter nucleus. So to put that in a context that perhaps a little easier to understand, if you were to line up all of the DNA in a human body end to end, that DNA would go from Earth to Pluto and back 3 times. But it's compacted all of us into us. So it's an incredible physical process that's intrigued me actually for many, many, many years. But this compaction has to occur in a way that allows the system to remain dynamic in order for it to be regulated. And we know that this organization of chromatin in the nucleus controls essentially all nuclear processes, and moreover that it goes awry in a variety of human diseases. So evidently it's functionally important. And if you look in textbooks, what you will see is this hierarchical model for chromatin assembly. You start with a double-stranded DNA. The initial compaction occurs through wrapping that DNA around an octamer of histone proteins to form so-called nucleosomes. Those then assemble into what's called the 30 nanometer fiber, which assembles into a series of higher order fibers, ultimately producing something like the mitotic chromosome. And while satisfying, the problem is that as imaging has gotten better and better over time, people just have not seen evidence for these hierarchical fibers in eukaryotic nuclei. Clearly, nucleosomes exist in nuclei, but these other structures really have not been to a large degree. And so the organization remains poorly understood. So we got into this problem by thinking about chromatin as an example of a multivalent polymer. It is an array of modular units, each of which is a nucleosome, which is obviously much, much, much bigger than a protein domain, but nevertheless, it is a module. The nucleosomes are wrapped by acidic DNA. The tails of the histones are very basic. And so you can think of this as an acidic system that could assemble through acidic-basic interactions. And so even though the scale is different from modular domain proteins, the linkers between nucleosomes or double-stranded DNA are much, much stiffer than polypeptide chains. But that essential feature, multivalency, is the same. And sure enough, when my postdoc, Ryan Gibson, was able to produce synthetic arrays of polynucleosomes, when we put them into physiologic salts, we see beautiful evidence for liquid-liquid phase separation. And we have a lot of data in this area. I'm not going to show you very much of it at all, but we think that this likely represents a basal state of chromatin compaction that is then acted upon by different modifiers to control a variety of nuclear processes. Now, one of the sort of most interesting things I think we've seen in this is that if we take two different kinds of chromatin, one of them unacetylated and one of them where the histone tails are acetylated, if we mix that with a protein that contains a multivalent reader of the acetylated histone tails, we can get two distinct phases of chromatin that interact with each other but don't coalesce. And so again, one of the problems in the chromatin field is that chromatin doesn't compact homogeneously. There are distinct, chemically distinct and functionally distinct regions in the nucleus. And this suggests that perhaps this process of liquid-liquid phase separation could contribute to forming that heterogeneity in the nucleus. Now, one of the things that has been particularly satisfying for me in our studies of chromatin is that it gets me back to structural biology after really a decade away from it. Because in contrast to the other molecules that undergo liquid-liquid phase separation, which are very small and can't be observed by cryo-electron microscopy, nucleosomes are much bigger. They're about 250 kilodaltons, and it's very easy in a cryo-electron tomogram, as I'm showing here, to observe individual nucleosomes. And so we've recently started to study our phase-separated droplets, and this is just one example of a cryo-electron tomogram that we've seen. What I hope people can see is there's lots of nucleosomes here that we can clearly make out. There are lots of different arrangements between them. One thing that's kind of cool is if you look hard, you can see that there are these examples of filaments that exist in this structure. These are not the 30 nanometer filament. They're only about 10 nanometers wide. It looks like a stack of nucleosome coins. That structure, in fact, has been observed in the nucleus. And so we think that by studying the structure of nucleosomes within these phase-separated droplets, we can gain significant insights into the organization of chromatin in cells. So again, this takes me back to my roots in structural biology, but now it's on a very different scale. We're not asking how are atoms organized with respect to each other, but rather how are nucleosomes as the key particle organized with respect to each other. So the last thing I want to mention here is my connection to the MBL. And you heard a little bit about this from Cliff. So in about 2012, Ron Vale and I started talking about setting up sort of this kooky idea of collaboration that we called the Hhmi MBL Summer Institute. And the basic idea that we then implemented also with Jim Wilhelm at UCSD was that we would bring together our groups and a series of other groups to work together in a common lab for about 8 weeks each summer for 5 summers in a row. And the idea was we could bring different people from different disciplines to study different aspects of biological phase separation over that period. And so Cliff had his picture of the 2014 Summer Institute. Here is mine. Here are all the people that were involved. You can see me, Ron Vale, Jim Wilhelm, and also I hope people will recognize Roy Parker, Hao Wu, Amy Gladfelder, and of course, Cliff. Tony was able to join us with his group of people for 2016 and 2017 as well. And so in addition to learning a tremendous amount, I think, of the early science in this field of biomolecular condensates, I think we also had some really sociological impacts on the field. It helped us frame the important questions in the field. And it helped establish something that for me, and I know for Cliff and Tony too, is very important, helped establish the culture of the field. as one that was very collaborative, very collegial, and really is multidisciplinary at its core. And I think that has really helped drive the science and make it a field that many, many people want to come into. And finally, it really established lasting relationships among senior and junior investigators in the field. Hence, in part, Tony, Clifford, and I have interacted so well for so long. So just want to finally also acknowledge the many, many people I've had the good luck to work with in this area in my lab. I won't run through all of the names here, but there are two that I do want to point out, and that is Pilong Li and Wei-Chin Cheng. Those were the first two graduate students in the lab that observed this phase separation process and were instrumental, key in making this connection to polymer chemistry and polymer physics that really drove our thinking in this project. So I thank all of these people for their wonderful work over the years in our science. And again, thank the Wiley Foundation, the Wiley Prize jury, all of the people involved for this wonderful recognition. Thank you.
Thank you so much, Mike. That was terrific. And thank you to all three speakers. Really astonishing stories. So we now have time for some questions. Again, if you have a question for any of the three speakers, please type your question into the chat room, and we'll try to get to it. I want to ask the first question, and it's an issue that has bothered me for a while. And that's the question of, is there a minimal size of a phase-separated entity? And any of you can answer this. So would you say that an entity that contains only 100 molecules cannot behave as a phase-separated entity? Does it have to be thousands, or where, you know? And of course, you understand that the reason why I ask this is because I work on telomeres, which are multivalent, but they're not that big. Cliff, Mike, Tony,
you want to kick it off, Cliff?
I did this. You know, this is I think a really interesting topic, because many of the structures that are in the cell doing important things are pretty small. They're more like 100 nanometers. We started studying the nucleolus because it was so huge it was easy to study, but things like the Pol2 transcriptional assemblies, I mean, these are really small. So it's a really interesting question. I can just say, from the perspective of the statistical physics and thermodynamics, the concept of a phase transition as something where there's a sharp transition as a function of concentration, temperature, and so forth, is only strictly defined in this thermodynamic limit of a large system. So there's something called finite size effects, and I was interested in this in the early days because exactly this question. And what happens there is you get smoother transitions. That is probably not relevant just for the cell. It's 10 microns or something is not quite there. But when you get down to these nanoscale, subregions where you're getting binding just locally, I think it is more relevant. The other thing, sometimes, if you can count the number of molecules on your hand or even 10 of your hands, that's not what we're talking about, right? But it's not, there's no specific number where you can say now phase separation is relevant, now it's not. And that's the challenge. And also, I think the most interesting physics going forward will be exactly in that space.
So I'll add one thing to that. I was just talking to another one of our colleagues, Ibrahim Sisse, yesterday about this issue. Ibrahim has looked at the density of molecules in some of the very small condensates, the 100 nanometer scale condensates. And what he sees is the density there is the same as the density of those structures once they become much, much, much larger. And so the implication there is that if density is a key component that determines functionality of these structures, probably the functionality that one observes in a larger structure probably will track down to the structures that exist in cells. But again, whether that is generally true or whether the effects that Cliff mentioned will change functionality at very small sizes is one of the exciting questions, I think.
Yeah, and I think I could say, teach you for that. I mean, as you get smaller, often there'll be other effects get very dominant, like polymer interactions. You know, as these condensates interact with DNA, that's going to strongly affect the physics in interesting ways. One thing sometimes I like to say is that, you know, phase separation is just a basic physical concept, which is just going to happen. It's like diffusion, right? And you know, and at the origin of life, there must have been, the cell must have had to grapple with this problem of everything wanted to phase separate. So I think you're asking, how dominant is it? It's like I like to use the thing of, like, say, reaction diffusion in the Turing models. Their diffusion was at the heart of the process. Well, when we talk about enzyme kinetics, even though you need diffusion, you know, we don't usually worry about diffusion too much. And so I think it's a matter of asking, there will be actually always sort of aspects of phase separation in any of these things, but how dominant is it in thinking about the biology is the way you can also ask that question.
All right, so there are actually more compliments about your talks in the chat room than actual questions, but we have some. And the first I want to call up is from Peggy McDonald. Peggy, can you unmute yourself and reveal yourself and ask your question?
Sure, those were all very interesting talks. I'm not in this field, but I was very curious about the concept of these phase condensated, liquid condensated things aging. And what is the molecular mechanism of that aging? And like if it's phosphorylation of something, why isn't that reversible? And could this be something that goes wrong in some of these diseases that involve these kinds of aggregates?
Yeah, this is also a question from Stephen Cohen, who doesn't have a microphone, has a similar question. I think perhaps Cliff, since you brought up the transition to gels and more solid states, maybe you can say something about it.
I can say something about that, but certainly I'm sure Mike and Tony have more to add. But you know, I think this idea that liquids can undergo a subsequent transition into solids makes a lot of sense from things even in the protein crystallography. You know, Dan Frankel and there's Peter Wright and all the papers going back suggesting that's kind of how protein crystals form or colloidal crystals. There's also a deep literature that I'm most familiar with in colloid physics on gelation. A lot of that was done by the Waits Lab and folks. This is part of a broader picture where people talk about glass transitions, which you can have dramatic changes in the material states, even while structurally, you know, it doesn't look any different. So you zoom in and look around, you know, you don't see anything particularly different, and yet there's a dramatic macroscopic change in the properties. And so I don't think we have a great answer for, you know, one picture yet. You know, sort of one of the things that certainly is relevant with the connection to pathological solids and this transition is amyloid fiber formation, which may be nucleated inside the liquid, you know, as low probability conformational changes to nucleate inside of a dense droplet. Obviously, the probability goes up quite a bit. Tony just had a really exciting paper suggesting this, what they refer to as an aging Maxwell fluid, more of a glass transition picture. They don't see, you know, he should tell us about that, but they don't see huge structural changes. Yeah. So I'll stop talking, but I think it's a really interesting question. And to my mind, it's still wide open.
Tony?
Yeah, I mean, I think Cliff gave a pretty good summary there. It is wide open. I would say that relating it to amyloids is interesting. The nucleation mechanism I talk about with tubulin is that, you know, cells must have to really pay a lot of attention to not form amyloids because we know how deadly they are. And phase separation, by raising the local concentration, could be allowing driving amyloids locally, either physiologically or pathologically. So that could be one way of relating the two together, these density transitions, on how they work. But as Cliff said, we don't know anything. Well, anything is too strong, but we know we're just scratching the surface of understanding what sort of post-translational modifications are driving these transitions, and no doubt that's going to be central to many of the disease processes.
So one of the things that none of us has spoken about at length here this afternoon is that there are many neurodegenerative diseases that are caused by mutations to molecules that are known to, in their natural form, exist in condensates in a liquid-like state, And mutations have been shown in vitro to cause them to drive into amyloid fibers. And so there's a sense that hardening that results from that effect, that transition from a liquid to a solid through that molecular mechanism, could be at least contributing, if not causative, in neurodegeneration.
Thank you.
So we have a question from Biomedical. Can you unmute yourself and show yourself to ask the question? Biomedical.
Sorry, I can't, he says.
Sorry, I can't.
Okay, he can't. Let's go to Rosanna Colapardo.
Ah. Hi, Rosanna.
Can you unmute and show yourself, Rosanna?
Maybe you can answer the question, Mike. It's, you know,
Rosanna and I are collaborating.
Oh, she said, Rosanna apologizes because she's with little kids. So let me try to, or can you see the question, Mike? Or Cliff? I think it's directed at Cliff.
Yes, two questions. One of them is to Mike, and it's on, oh no, that's just a great talk.
Yeah.
Right. I guess the question is to me,
Yeah.
Mike, is there a likely hypothesis for a general function of the multi-phase organization of condensates? Are multi-phase condensates widespread? I do think that they're widespread. You know, the Nucleolus is the clearest example. Again, probably we've been able to dissect some of that because it's large, but we had a paper last year looking at stress granule organization and stress granules themselves together with p-bodies, to which they are, you know, often physically juxtaposed, similar components, but compositionally distinct and not mixing. There's other examples where things look like, you know, multi-layered structures. Joe Gall has this beautiful image that I show sometimes from the Xenopus germinal vesicle with a Cajal body and a snurposome, which is a speckle-like assembly. They look like Mickey Mouse structures, even though there's no membrane. I think those things are everywhere. It's also not surprising. There are some fundamental relationships between the number of molecular components in a system and the number of distinct coexisting phases that there can be, and it's like n-2 or something, this relationship. So I think that it's probably very likely that you know, all the things we're talking about are coexisting liquid phases. Just in some cases they're sitting next to each other. In some cases they're one inside the other. And the determinants of that have to do with the surface tensions, and those surface tensions have to do with the biomolecular interaction energies. So I think we have a framework for that, and it's in terms of function, I like the idea, and that's what we think about the Nucleolus, that, you know, you have the sequential processing in the different layers, and that may be something that's more general as well.
Yeah, I do want to add that I think that, you know, that work of Cliff's from his lab on multi-phase is probably one of the most important things that will come out of this, in my opinion, in the long-term condensate field, because the old concept of channeling in enzymology, you know, where the product of one becomes a substrate next, no one ever really understood that. But that multi-phase system, I think, could provide a really important mechanism.
Great. I think we'll round it off here. It's 2:30. Amazingly, we've had around 240 participants throughout, but they're leaving now. So I think we should probably end this discussion. This is really terrific. Beautiful talks, deep and inspiring and interesting discussion. And I want to end with thanking Debbie Wiley and the Wiley Foundation again for allowing us to celebrate fantastic science this way. Thank you all very much for your participation.